I think people are coming back from breakout rooms. I think Suzanne, you're up next, right? You're ready for that?
Yes.
Perfect.
Hopefully.
And okay, let's see a bunch of people joining. So, and just a note to everybody, I'm just going to quickly share my screen while we're waiting. So, looking on actually any of the sites, but let's go to ViewPathDB. Again, if you're trying to find the workshop page, go to learn how to use ViewPathDB. And I know somebody provided the link, the direct link in the chat. We can do that again. Go to workshops, select today's workshop, and now we have the table. And now finally, these are updating. Something was preventing the updates this morning. It was actually from a user error from my end, but now it's fixed. So the exercises all are showing up here for today, and I'll upload the exercises for tomorrow, either late tonight or tomorrow morning before the workshop. And so we'll be able to access them directly from this workshop page. If you click on any of these links, it'll download or load the PDF in your browser, depending on your configuration. All right, and I see Uli shared it with everybody. Okay, Suzanne, I think we'll turn the show over to you, unless any of the instructors, anything, not to take too much time from this portion, but is there anything really essential or burning that you wanted to make sure gets mentioned here to the entire group? Okay, Suzanne, all yours.
Okay, sounds good. I have shared my screen. Do you guys see PlasmaDB? Yeah.
Yes.
And
perfect.
Do I have to start record? No.
No, I think it started already.
Okay.
Rachel did that already.
Yep. All right. So we just spent a few minutes getting familiar with the site search and the data on the gene page. And now we're going to focus on understanding our specialized searches and our search strategy system. I'm going to stop my video because that freaks me out. And the difference, I just want to mention the difference between the site search and our specialized searches. So the site search is a text search, right, that queries all pages everywhere on our site. So everything that you get back is you get a gamish of different record types back. But there are text searches that return only genes, right? And this text search queries only the gene annotation tables in our database to return only gene attributes. So there's a very difference. This is these searches in the search for menu, search functional data associated with these specific record types one at a time, whereas the site search will do all at a time. Okay. Now, to understand these specialized searches and the search strategy system, I'd like to emphasize the different data types that we have. And that's best shown on the gene record page. Right, so here I am at PlasmaDB on the gene page for apical membrane antigen, which compiles all of the database data for this single gene. It's essentially, as you just saw, a categorized list of data. And so from the genome sequence and annotation, we know the gene structures, introns, exons, UTR regions. We know the genomic location. And these are all coordinates on the genome. So this information comes to us in the form of numbers, if you will. Many of these genes have product descriptions, which is information in the form of text. But the presence of a product description then depends on the maturity of the genome annotation. Some genomes have not been studied as well, and so have much less information attached to each of these genes. And that's not to say anything bad about that organism or the people studying it, just some of them have not been studied as long or are more difficult to culture, so are not amenable to different types of experiments. And then Another example is that as we bring in the genomes, we determine orthology across all of the genomes, and that is information that is an association between genes and organisms. We also integrate different population studies, right? And this data consists of summary data from our analysis that is numbers, and then individual outputs of certain single nucleotide polymorphisms, coordinates for that. So, and a couple different types of data addressing polymorphisms. And then last but not least, I'll show you the transcriptomics data. We have expression data in the form of transcriptomics or proteomics. Some are expression or abundance values, and other data is present absent calls, such as peptides in a proteomic study. And for, you know, like each of these is a different experiment. And we have many different types of experiments. Some are time courses with a 1 or 2 hour resolution. Some just compare life stages. Others look at knockout mutants, right? So lots of different data. And not only do we have this data for this gene, but we have it for every gene in this organism. And every organism in this database, if that data exists for that organism. And then we have this type of data for all organisms that we support, which is upwards of 570 organisms now. So imagine a set of tables with all of this data. If you were a computer scientist, you could write a script that pulls out specific portions of this data. You could say, show me all genes that have 3 exons. Show me all genes that are expressed in this certain life cycle stage. Show me all genes that are orthologs of these other genes. And that's what our searches are. Scripts that access our pre-computed omics data and return results based on your parameter choices. The gene pages access the data for a specific gene, and the searches access data across all genes, all organisms, and data types, if you use the strategy system well. And then we've built ways for you to combine the results of individual searches to find groups of genes with your desired characteristics. Even if you have like 5 characteristics, you can string 5 searches together to find meaningful relationships between, come up at the end with a single set of genes that you know have certain biological characteristics. So in the upcoming example, we're going to pretend that we are all Plasmodium vivax researchers who are interested in the egress of gametocytes from the infected red blood cell. You could imagine that the parasite would need some enzymes to help it break up the red blood cell wall. You could also, and let's also say that we're interested in the evolution of this process. We can build a strategy that finds genes that are proteases, that are expressed at the gametocyte stage, and that carry non-synonymous mutations. Okay, so let's talk about the search form. Omar went over this a little bit. We can access it here. I'm gonna do a text search. You can also access it from this menu up here. So the search page consists of the parameters that define the query that you're going to run. Then down at the bottom, there's a description that with help information. Sometimes this is very, very useful. And then we have links to the data set records that describe the data that this query accesses. Okay. So we're going to run a search, text search for the word protease. And this query that our computer scientists have written has 3 parameters. We need to know what organism that we want to query, and I'm just going to say select all. We need to know the word that we're looking for, and I have already typed in here protease. And then we need to know where to find it. So we've actually looked at all of our tables and said, well, there's textual information in all of these places. And so we've allowed you to, select to look in only the product description and the PDB chains, or you can just select all and look for them all there. So I'm going to do that and click Get Answer. And I am taken to the My Strategies section, where my result appears at the top as a graphic. So this is my search strategies. And I can always get to that from any page by clicking this My Strategies up here. So this search returned 6,203 genes. They are distributed across all of these organisms that we searched. And I can see that in the organism filter, and I could use this to show only one organism up here. And then to the right of that is a table of all of the IDs that were returned. And there's a few things you can do with this. Omar has already described. I can add any of the data that was on the gene page, more or less. I can add as a column here. If it's suitable as a column, I can download my list, all kinds of things. So that's how you run a search. And what the results look like. All right, so we have our first search, and I'm going to demo 2 things. One is how to combine searches, and the other is how to transform between organisms. Now, we want to run another search because we definitely want these protease enzymes, but we also want, say, gametoside expression. And when we go to run another search, we're going to get a second list of genes. Not only are we going to have to fill out another form, but we're going to have to say how we want to combine those results. So let's talk about that a little bit. I have this. All right. So when we get, when we have 2 lists, we'll have a list 1 and a list 2, and they're both filled with IDs. And mathematically, there's only a couple things that you can do with that, right? You can intersect those 2 lists and say, show me the IDs that are common between these 2 lists. You can union them and say, show me all of these. Give me another list that has all of these. You could subtract them, say, which would be to ask, if you did 1 minus 2, you'd say, show me all of the genes in here that are unique, that do not appear in this list. And you can go vice versa, 2 minus 1. And then we also have this other tool called collocation, which allows you to ask the question, show me all genes in this list that are close to, on the genome, these other genes. Okay, let's delve into these a little bit more. Yeah, let's talk about intersect a little bit, because the way you combine searches determines the biological characteristics of the genes in your combined result, right? So here I ran 2 searches. I ran a search that returned genes with signal peptides. And that's all I know about these genes, because that's the only question I queried right now. They may have other attributes, but for sure they contain a signal peptide. And then I ran a second search for genes with transmembrane domains, and that's all I know about these guys, right? If I choose to intersect them and find genes that are in common between the 2 lists, then my result of that combined result are genes that have a signal peptide and a transmembrane domain. On the other hand, if I was to union them, I would end up with all three types of genes. I would end up with genes that have signal peptide and transmembrane domains, but I would also have, in my result, transmembrane containing genes that do not have signal peptides, right? Those are these, and signal peptide containing genes that do not have transmembrane domains, and that's these. Okay, so you have to think sometimes about the way that you are combining these, your results, and what you want to end up with. For the 1 relative to 2, which is our co-location tool, it's very powerful, but can also be a little tricky to use sometimes. So we need the co-location tool for, well, this is one of the examples of what you need the co-location tool for, and that is if lists do not contain the same type of ID, right? If I do a search and I get gene IDs in the first one, and I do a second search for single nucleotide polymorphisms, I cannot combine those these results with intersect or plus or minus or union, because there are no IDs common to these groups. It's impossible to intersect those groups, these two lists. There are no gene IDs in this list of SNP IDs. You just can't do that. So we do know, however, the genomic location of each of these entities. We know where the genes are, and we know where the single nucleotide polymorphisms are. So we have built a tool for asking, show me all of these genes that are close to these SNPs, that have these SNPs in their upstream 500 base pair regions, or show me all of these genes that contain any one of these SNPs. So that's our co-location tool in a nutshell, and you will do an example of that in this exercise. And the last thing I wanted to demo was an orthology transform. I can't recall if Omar showed this, but so here I have my text search that I just ran, and
I did not show it, just so you know. I did not show
okay,
it.
Great. Yeah. I just ran this text search, and I searched all organisms, so I have a gamish of these are all gene IDs, but they're all from different organisms. I'm going to go ahead and filter that and show only 3D7 genes, and it looks like I'm going to come up with 833. I'm going to apply that filter, and that is represented then in my graphic up here. I have only 833 genes, and if I had 4 or 5 searches and I filtered this first to show only 3D7, then that is going to be sort of define the first combined result. Okay, so I have only these are PDF 3D7 genes that contain the word protease in their records. And say I'm interested in Vivax genes, I can say add a step, transform into related organisms, related records. There's 3 choices. I'm going to choose orthologs because I want to go to pvivax. Then I choose my organism that I want to convert to and say get answer. And it turns out that these 833 protease, suspected protease genes correlate with 872 genes in pvivax. Now that is different, right, from searching first off for the word protease in pvivax records. Let's go ahead and do that. I'll do a text search only in P-Vivax, select only this for the term protease in all of these fields. I'll click Get Answer. And I have 112 genes. That's not a wrong answer, right? The basis for the difference in these is that the annotation for P-Vivax is not as mature as the annotation for PF3D7, where many genes are tagged with the term protease, but here fewer genes are tagged with the term protease. So these, the transform by orthology is a great way to make inferences from organisms that have a lot of data to organisms that might not have so much. And that's the last thing I wanted to demo. I think we can, if there's no questions, you can go to workshops. And the search strategies exercise is linked here. We can go to that. And this, I'm not sure we'll finish it all, but we can give it a good try, right? So there's a lot written here that it's intended to be sort of a self-taught thing. So if you are interested in going back and reading all of this for a little bit more in-depth description of what you're doing, that's fine. But on page 4, starts in with the exercise. And
Great,
you
thank
will,
you.
and, yeah,
Go ahead. No, I was just going to say thank you, Suzanne. This is good. So we are a little bit ahead of schedule, which is great. Which means that that we can start this exercise early. So Rachel, you can put us in our breakout rooms. Note that we will stop the breakout rooms around about an hour from now. And then we will have a final wrap up. We'll just discuss, the day and what we're going to be doing tomorrow. And then, as I mentioned earlier, in case you missed it, you're welcome to leave then at the top of the next hour. So at the top of 1 p.m. US Eastern time. And then, but we'll keep the Zoom link open for another half an hour in case you have some burning questions you want to stick around and ask them. Some of the instructors will stay on for you. Okay, Rachel, you can go ahead and open up the breakout rooms. Thank you.